Hello, welcome to the first part of Lecture 10, How to Build a Protein. Build a protein? What you talking about? Well, there are four biomolecules of life. I believe I have said this before, but if not, there are four of them. We've talked about nucleic acids just a little bit when I introduced GMOs. The second is proteins, and the third and fourth are carbohydrates and lipids. We're going to get to those later in the semester. Yeah, funny dog. What do you call a professional teenager? A protein. Okay. Well, that's not actually the definition of protein that we're going to use in the class. They're polymers that are made up of amino acids. These are the monomers, or the individual molecules that have a similar chemical motif that is going to form these long chains that become proteins. An amino acid, if you were to break that word down, you would understand that it has both an amine group, or a nitrogen-containing group on one end of it, and a carboxylic acid on the other. So that's why it's called an amino acid. The alpha carbon of the carboxylic acid, so the next carbon down the chain, is attached to the amine. So all amino acids have this basic structure that you see here. There's going to be a nitrogen, an alpha carbon that has a side chain that is going to vary. There's going to be roughly 20 different side chains that we can identify that make up the proteins in our body. And then, and this is what makes each amino acid unique. And then, of course, the carboxylic acid on the other end. So this backbone is nitrogen, carbon, alpha carbon, carboxylic acid with different side chains. Oh, the dog is back. What do you call an acid with an attitude? Amino acid. Yeah, funny dog. Here they are, the 20 common amino acids as you would draw them at physiological pH. This is the first half of them divided by, so you can divide the 20 amino acids up into four categories based upon their physical properties. The first is hydrophobic amino acids that are nonpolar. And so there are 9 of them. Glycine, alanine, valine, leucine, isoleucine, proline, phenylalanine, methionine, and tryptophan. And they have a pretty large diversity in terms of their side chains, but all of those side chains are fairly non-polar molecules, meaning there's a lot of CH, a lot of carbon, a lot of hydrogen, carbon-hydrogen bonds that you find in these. There are the 3 branch-chain amino acids, valine, leucine, and isoleucine, which are important for muscle building, glycine, which is important for forming collagen. Tryptophan is the precursor of a number of molecules in your body, including serotonin and melatonin. And then there's this other category of amino acids, the hydrophilic or polar amino acids. They're not charged, but they are polar. So you'll notice on them, in them, that there are a lot of hydroxyl groups such as serine and threonine, a lot of thiol groups such as cysteine. The SH is pretty similar to the OH. There are a lot of carbonyls, like in glutamine, asparagine, tyrosine has an OH as well. So these are hydrophilic or polar amino acids that are going to be able to often participate in hydrogen bonds. These side chains can participate in the hydrogen bonds, but they are not charged. You'll notice in all of these, the backbone is the same. It's NH, NH3, CH, CO2 minus. So NH3 plus, CH, CO2 minus. All right. This is what they would look like at physiological pH, floating free in solution. 2 other categories are also both hydrophilic. One of the categories are hydrophilic molecules that aren't, the side chain is charged in a basic fashion, meaning that they can act as proton acceptors. So these are lysine, arginine, and histidine. And then there are acidic amino acids that can act as proton donors, and those are aspartic acid and glutamic acid. Actually, those are shown in their ionized form as the conjugate base. So that would actually be aspartate and glutamate. But those carboxylic acids that you see can remain protonated in different at different pHs. All right, so those are all the amino acids. Let's make a protein. There are several stages to consider when talking about protein structure, and the first of them is primary structure. That is just the linear sequence that the aminos make when they form a chain. Well, how do they form a chain? That is the formation of the peptide bond. So the amide bond that is formed between one end, the NH3 end of the protein backbone, and the other end, the carboxylic acid or CO2 minus end, that bond is a amide bond, chemically speaking, but within protein chemistry we call it a peptide bond. And when you link two peptides together, we call that a dipeptide. It's not quite a protein yet, but it's a dipeptide. So let's take amino acid 1. Who knows what it is? The R group can be whatever you want of those other 20 amino acids that we just looked at. As likewise with amino acid 2. It doesn't really matter what those side chains are. They can link up with one another. And this is a type of dehydration reaction where you can notice that we're removing a water molecule as we form this peptide bond. So the backbone is now NCC-NCC. And we can extend this chain on either end with more peptides. So the primary structure of a protein is simply the linear sequence of amino acids that are all linked up in a chain. So this primary sequence is given using 3-letter codes in the DNA, and a polypeptide, like a very small protein, has something less than 100 amino acids in a row. So here is a very small protein, a polypeptide called cytochrome C, and cytochrome C has 104 amino acids, and I've color coded them in this picture to show the nonpolar or hydrophobic amino acids. There's 47 of them. There are 24 polar amino acids, or polar neutral or hydrophilic neutral amino acids, and then the acidic or basic, the ionized amino acids are in red. So it's cytochrome C. These peptide groups are going to form the backbone of all proteins. Any protein that's out there is going to have this NCC, NCC, NCC repeating motif with the peptide bond between the carboxylic acid carbon of one amino acid and the amino group of the subsequent, forming what's called an amide or peptide bond. And then the different side chains can stick out from that backbone. So here's some cartoon on the left of a peptide, long chain in blue. The backbone is in blue. Side chains, different side chains are described in red here, and we're going to zoom in a little bit, and we see this in this box. There is, in this segment, we have alanine, aspartic acid, and alanine. So an alanine, aspartic acid, and an alanine, 3 of them in a row, and we can see their different side chains sticking out from the backbone. All right. On to secondary structure. Secondary structure is, there are these structural elements. There's only 2 types, really, of them. the alpha helix and the beta sheet. It is in like higher order protein studies, structural biology of proteins. There are some other subcategories here, but basically alpha helix and beta sheet. So an alpha helix is a tubular spiral. It's a long strand where the peptide backbone forms this coil that extends in a linear fashion, and it's held together by hydrogen bonds that form between the hydrogen on the nitrogen of one in one amide group, and the carbonyl, the CO of another amino acid that is down the chain, about 3 amino acids down the chain. And this allows for this coil or spiral to have quite a bit of stability, and they can act as sort of like the girders of a protein. They can act as like long, stable girder-like structures. On the other hand, we have beta sheets, and there are these long sheets that also have a similar hydrogen bonding pattern, but it's not in a spiral. It's these sort of you can think of it more like corrugated metal for a roof or something like that. There is these lines of the peptide backbone goes in one direction, and then another part of the peptide backbone travels adjacent to it, and you can get this hydrogen bonding that forms these sheets that can also act as a very stable structural motif in proteins. Proteins have secondary structures in them, but they are not entirely described by these secondary structures. They're just sort of a basic three-dimensional motif that the protein can have. So these secondary structures can add together and form tertiary structures. And tertiary structure is the actual sort of global shape of the protein. And tertiary structures break down into 3 categories. You can have fibrous, long, narrow, water-insoluble proteins. So, for example, hair, silk, tendons, ligaments are made up of these fibrous proteins. And like when you're cooking meat, for example, it is collagen is a type of fibrous protein that is making up the connective tissue that's holding the muscle fiber together. And so breaking down those fibrous proteins is part of making that meat more tender. The second category are globular proteins. So these are proteins, as the name would suggest, that are big globs, large globules that are folded into compact shape. So most of the proteins that dissolve in solution, that are water soluble, are in this category right here. And these proteins have a mix of polar and nonpolar amino acids. We'll see how that works in just a bit. And then finally, there's a third category of proteins that are membrane bound, and those have very specialized functions that we probably aren't going to get to talk about a whole lot, but they're proteins that are embedded in the membrane of our cells. All right, so to help you understand all this, I'm going to switch gears and talk about knitting for a couple slides. Yeah, it better be good. Hope so. So I don't know if anyone's a knitter out there, but if you've ever followed a knitting instructions, first you have to follow the primary structure, right? The primary sequence structure. And that's just the description of each individual stitch and how to link each stitch up together. If I were to show you this primary structure, row 1, I can't even read all that. Those are the instructions for something. If I were to read that, I don't really get a sense of what that is. I'm not really sure what I'm looking at there. But it is sufficient to make whatever that is that I would be trying to knit. Then there is the secondary structure. All right, so as I do this, each of these rows, it has a unique structure. There's a purl versus a knit. There are these like cable stitchings. There's this sort of meshwork, and these different secondary structural elements begin to appear from the knitting. I still can't really tell what I'm looking at here in terms of the overall structure. Whereas the tertiary structure describes the three-dimensional shape of whatever it is that I'm making, and this person is wearing this shawl or scarf or whatever it is there. That would be analogous to the tertiary structure of a protein. There actually is the possibility for quaternary structure or fourth-order structure. This is when two or more polypeptide chains or proteins form a cluster, and that cluster, they were created separately, but they come together for whatever that protein is to be fully functional, and so we would call that a quaternary structure. So a little bit about the forces that hold proteins together, and then we're done. I've shown you, we've seen that these linear chains can have hydrophilic amino acids mixed with hydrophobic amino acids along its length. When that protein, that linear chain of amino acids, goes to fold itself, the hydrophobic amino acids are going to gather together to minimize their interaction with water because they don't like to be around water, and water agrees it doesn't really want to be around them. And then that protein is going to align all the hydrophilic amino acids, and however it folds itself up, on the outside to be near the aqueous environment. All of this is important to consider because when we have to understand how proteins are put together if we're going to talk in any kind of real way about how we take proteins apart in terms of cooking or otherwise denaturing them. Here are some other forces that hold a properly folded protein into its native state. All right. So there's a couple categories here. You can have charge-charge interactions or ion-ion interactions, such as this interaction between aspartic acid and lysine. So we have an NH3 plus on the lysine and a CO2 minus from the conjugate base of aspartic acid that are attracted to one another, and they can hold two arms of, for example, two different alpha helices together to give the protein the structure it needs. We can have hydrogen bonds between amino acids. So here are two serines that are able to form hydrogen bonds with one another. Also adding a little bit of glue holding these two alpha helices towards one another. You can also form what are called disulfide bridges between two cysteines. And this is an extremely powerful form of side chain interaction because it's forming an actual covalent bond between the two sulfurs in the cysteines. That's a very powerful interaction and difficult to break. So each of these different interactions, the charge-charge interaction, the ion-ion interaction, the first one there, pretty strong, sort of moderate strength. The hydrogen bond, less so, but it can have additive strength if there's a lot of them. It also has a lot of degrees of freedom, which can give certain properties to the protein that it might need. The disulfide bridge, very strong interaction because of the formation of a covalent bond. Each of these is going to be susceptible to different types of cooking and different types of other culinary techniques to break them, and it's going to give the foods that have these different sorts of interactions in them different properties when they have been cooked or heated or otherwise manipulated in the kitchen. Thank you for tuning into that part. Part B is up next. How we built a protein, Now we're going to talk about how to break a protein down. Thanks.